Microbial metabolism is broadly divided into catabolic and anabolic reactions, which together constitute the cell's metabolic network. Catabolism involves the breakdown of complex organic molecules into simpler compounds through hydrolytic reactions, releasing energy in exergonic processes. This released energy is conserved primarily in the form of ATP or reduced electron carriers such as NADH and FADH2. Catabolic pathways are typically oxidative and generate metabolic intermediates that feed into biosynthetic pathways. Anabolism consists of biosynthetic reactions that construct complex cellular molecules such as proteins, nucleic acids, lipids, and polysaccharides from simpler precursors. These reactions are endergonic and require energy input, usually supplied by ATP hydrolysis or reducing power from NADPH. Dehydration synthesis reactions dominate anabolic pathways, forming covalent bonds between monomers. ATP serves as the universal energy currency of the cell. The energy stored in ATP resides in its phosphoanohydride bonds, particularly the terminal phosphate bond. ATP synthesis is driven by redox reactions in which electrons are transferred from reduced substrates to electron acceptors, releasing energy that is conserved through phosphorylation mechanisms. Many metabolic pathways are amphibolic, meaning they function in both catabolism and anabolism. Central metabolic pathways, such as glycolysis and the Krebs cycle, not only generate energy but also supply precursor metabolites for biosynthesis, allowing efficient integration of cellular metabolism. Metabolism. Catabolism. Exergonic reactions. Hydrolysis. ATP production. Electron carriers. NADH and FADH2. Anabolism. Endergonic reactions. Dehydration synthesis. ATP consumption. NADPH use. ATP. ADP plus inorganic phosphate equals ATP. Energy from redox reactions. Amphibolic pathways. both catabolic and anabolic.